Hello students, welcome to Read Med Prep Academy channel. Today in Principles and Processes of Biotechnology-Part-V, we are going to talk about applications of biotechnology, interdisciplinarity fields of biotechnology and new vectors in biotechnology. Biotechnology is one of the most important applied interdisciplinary sciences of the 21st century. It is the trusted area that enables us to find the beneficial way of life. Biotechnology has wide applications in various sectors like agriculture, medicine, environment, and commercial industries. Some of the applications of biotechnology in the production of transgenic plants, human insulin, bioproduct synthesis, biochip manufacture, genetic engineering, single-cell protein, secondary metabolites, environmental biotechnology. Let us discuss one by one. Transgenic plants. This science has an invaluable outcome like transgenic varieties of plants. Example, transgenic cotton, Bt cotton, rice, tomato, tobacco, cauliflower, potato and banana. The development of transgenics as pesticide resistant, stress resistant and disease resistant varieties of agricultural crops is the immense outcome of biotechnology. The transgenic plants and animals, some of the examples: golden rice, rice modified with daffodil genes to have more beta carotene, which the body converts to vitamin A. Flavor saver tomatoes, which are modified by the removal of genes responsible for the softening of the fruit, meaning the tomatoes spoil more slowly. Bt corn, corn modified with the bacterial insecticide gene so that it produces insect toxins with its cells, protecting it from pest species. Aqua advantage salmon, salmon modified with growth hormone, regulating genes in order to grow to market sizes in significantly less time. Glow in the dark animals, animals are modified with genes for fluorescent proteins which will glow in the dark. This novel feature serves no practical purpose. Human insulin, the synthesis of human insulin and blood protein in E. coli and utilized for insulin deficiency disorder, diabetes mellitus in humans, is a breakthrough in the biotech industries in medicine. How the human insulin production is done? You can see that the human pancreatic cell is taken with the DNA and the human insulin producing gene is separated, and a bacterial DNA which is of the plasmid type is taken and the plasmid DNA is cut with the restriction enzymes, and the human insulin producing gene is incorporated into the plasmid DNA and a recombinant DNA is formed, and this is introduced into the bacterial cell and is allowed to multiply in clones in the fermentation tank. And this bacteria which multiplies produces large amount of human insulin which is extracted and purified and is manufactured in the vials for human use. Bioproduct synthesis, the synthesis of vaccines, enzymes, antibiotics, dairy products and beverages are the products of biotech industries like the pharmaceutical manufacturing, baking, dairy, beverages, feed, biopolymer, paper and pulp, leather, textile, cosmetics, detergents manufacture, organic synthesis, and also in waste management. Biochip manufacture is based on the biological computer, is one of the successes of biotechnology, where it has a central semiconducting molecule surrounded by a protein frame which is placed on the protein support. Multiple biochips are arranged in a microarray which is attached to the transducer and a signal processing unit which brings out the output in the computer. The genetic engineering involves genetic manipulation. Tissue culture involves aseptic cultivation of totipotent plant cell into the plant clones under controlled atmospheric conditions. How the genetic engineering is done? The bacteria is taken and the DNA is extracted and isolated from the bacteria, and the cloning and designing of the genes is done, and transformation is done and is introduced in the bacteria again, and the cells and the tissue culture is done. These cells of the bacteria are introduced into the plants and the plants are allowed to breed themselves, causing excessive commercial production of plant breeding. Now, single cell protein. Single cell protein is extracted from spirulina, which is utilized in food industries. Spirulina is a microscopic algae and well accepted food world over. A simple one cell form of blue green algae that thrives in the warm fresh water bodies. It contains the most remarkable concentration and combination of nutrients known in any food, plant, grain or herb. It contains 65 to 70% of protein. Now, secondary metabolites, the production of biofertilizers, biopesticides and enzymes as secondary metabolites. These secondary metabolites are used in the agrochemical industries, cosmetic industries, insecticides, flavor producing agents, natural dyes, raw materials for industries, food coloring agents and in pharmaceuticals. Environmental biotechnology is helpful in the production of biomass energy, biofuel, bioremediation, phytoremediation for environmental technology. The uses of biomass energy for producing heat energy. Anything from the nature which can burn to heat. Example, charcoal, wood, mustard oil. For producing electricity using method is same as oil. Burn it and get energy either for a state or a house. Example, wood, crop residues, mustard oil. Biofuels. Liquid biofuels, bioethanol. Ethanol is a form of alcohol which is fermented and distilled from a wide range of plants like such as wheat, corn or woody material. Biodiesel is produced by chemically upgrading oils obtained from pressing of the oil plants, so gas biofuels. Biogas. Synthetic natural gas is also obtained. Solid biofuels from wood, charcoal and biomass pellets. What are the uses of biofuel? As an alternative cleaner fuel source, as power vehicles, heat homes, cooking and for power production, as replacement for fossil fuels for home and transportation purposes, as substitute for toxic products used for paint removal, as home heating oil in domestic boilers. What is bioremediation? Bioremediation is the process of using organisms to remove or reduce the pollutants from the environment. Bioremediation is the use of biological systems for the reduction of pollution from air, aquatic or terrestrial systems. The process involves aerobic or anaerobic microorganisms that results in degraded products. Bioremediation is used to reduce or remove hazardous waste which has already polluted the environment. You can see on the right side, the microorganisms are used to remove the pollution from air, water or soil, and the degraded product are completely destroyed by detoxification, which is eco-friendly. Or sometimes the degradation can be incomplete or hazardous, like the production of tri or tetrachloroethylene or vinyl chloride. Phytoremediation is the use of certain plants to remove contaminants or pollutants from the environment like the soil, water or air. The green technology, pollution-free technology in which pollution is controlled at source. Now let us move on to interdisciplinarity fields of biotechnology. Biotechnology is one of the most important applied interdisciplinary sciences of the 21st century. It is the trusted area that enables us to find the beneficial way of life. Biotechnology has wide application in the various sectors like agriculture, medicine, environment and commercial industries. Here you can see a sum up where it is used in genetics, the science of heredity and variation in organisms, in molecular biology, study of biology at molecular level, in immunology, study of body's defense mechanism, in microbiology, study of microorganisms and their effect in humans, study of cells and their interaction with environment in cell biology, in biochemistry, the study of chemical processes of the living systems, bioinformatics in the management of biological information with the help of computer technology, biophysics, application of physical principles and methods to biological problems, nanobiotechnology, which is the nanotechnology with biological and biochemical applications, Biomedical Engineering, applications of engineering principles and techniques to the medical field, tissue engineering, study of the biomaterials and stem cells to repair or replace the body tissue organs, chemical engineering, the conversion of raw materials into valuable forms with the application of physical and life sciences. In crop technology, a cultivated plant grown commercially in a large scale. In diagnostics, characteristics or indicative of a disease. In pharmaceuticals, drug or medicine used in medical treatment. In vaccine manufacture, any antigen or antibody that confers immunity to a particular disease. or livestock, domesticated animals kept to produce commodities or favorites. So let us discuss the different fields of biotechnology. Here in genetic engineering, we know that the insecticide gene created using recombinant DNA technology, where the required gene is cleaved from the DNA and a plasmid vector is taken and that vector is also cleaved, and the cleaved DNA from the DNA strand and the plasmid are joined together to form a recombinant DNA, and they are allowed to grow in the petri dishes in the culture media in the laboratory, so that the clones of DNA are formed, and these are selected for insecticidal cells. The cells are used for the plant propagation. Hence, the insects that feed on the plants will die and the insect-resistant plants are obtained, otherwise called transgenic plants. In molecular biology, which is a branch of biology that deals with the molecular basis of biological activity, chiefly concerns itself with understanding and the interactions between the different types of DNA, RNA and protein biosynthesis, as well as learning how these interactions are regulated. In immunology, various types of immunotherapies used in the form of bacteria, oncolytic virus, cytokines, lymphokine activated killer, LAK, and natural killer cells, NK cells. Then T cells, tumor infiltrating lymphocytes, dendritic cells or mesenchymal cells, CAR cells, fusion proteins, monoclonal antibodies in the manufacture of vaccines. How biotechnology is used in microbiology? Microbiology is the study of microorganisms like microbes. As we know, they can be harmful or they can also be helpful. Microbes can be used for breaking down the harmful waste, recycling of nutrients within the ecosystem, food production like cheese, alcohol, bread, yogurt, etc. In gene therapy, uses a virus or bacteria to enter a cell and replace the defective genes or create substances to treat disease. In cell biology, the drug discovery and development has been used. Growing stem cells in cultures to observe the earlier signs of a disease. Implants and transplants for treatments. Introduction, the reprogramming proteins directly into the body to stimulate stem cells in their natural niches. Somatic cell therapy, cell and tissue engineering. In biochemistry, it is the mainstay where the biotechnology is used in genetics, biology and metabolic biochemistry. In bioinformatics, the application is the knowledge-based drug design, forensic DNA analysis and agricultural biotechnology, computational studies of protein-ligand interactions, knowledge of three-dimensional structures of proteins. This informatics-based approach significantly reduces the time and cost necessary to develop drugs with higher potency, fewer side effects and less toxicity than using the traditional trial and error approach. In forensics, results from molecular phylogenetic analysis has been accepted as evidence. Genomics and bioinformatics are now poised to revolutionize our healthcare system by developing personalized and customized medicine. In biophysics, it's an interdisciplinary science that applies approaches and methods traditionally used in physics to study biological phenomena. Biophysics covers all scales of biological organization from molecular to organism and populations. Biophysical research shares significant overlap with biochemistry, molecular biology, physical chemistry, physiology, nanotechnology, bioengineering, computational biology, biomechanics, developmental biology, and system biology. What is nanobiotechnology? Nanobiotechnology is the creation of fundamental materials, devices, and systems through the understanding and control of matter at nanoscale, 1 to 100 nanometers, where few functionalities and properties of matter are observed and harnessed from a broad range of applications. An engineered DNA strand, you can see in the lower left. In the middle, you can see the pRNA, tiny motor. On the right lower side, you can see the semiconducting metal junction formed by two carbon nanotubes. What is the application of bio-nanotechnology in various sectors? Nanoviricides, tissue engineering, molecular imaging, gene delivery, targeted drug delivery, biosensors and biomarkers, food and agriculture, DNA nanotech. What is biomedical engineering? Bioengineering is equal to biomedical plus biotechnology. Bioengineering is a discipline that applies engineering principles of design, and analysis to biological systems and biomedical technologies. Bioengineering is the branch of engineering in which the engineering principles are applied to define and solve problems in biology and medicine. Application of biomedical engineering is the engineering principles to living structures such as creating artificial organs, chemicals, drugs and tissues. Here you can see the biomaterials that have been used in aneurysm repaired with the synthetic graft material. The tumor growth and metastasis shows that there are tensile forces and compressive forces and shear forces that are studied by biomechanics. Biomedical modeling is done where the model of compression in knee joint can be done in different modules. Here you can see the biomedical devices like the cochlear implant where the sound processor, transmitter, receiver and cochlear implant electrodes are used in patients for impaired hearing. The progress in tissue engineering helps in engineering materials like the novel chemistries, growth factors, biomechanics, used in engineering tissue architecture like decellularized organs, 3D printing technology, self-assembly, in engineering cells like the CRISPR and Cas9 that have been used as the genetic tools, adult stem cells, iPSCs, and cell source. What is the chemical engineering and biotechnology relationship? The chemical engineering has a pivotal role in the delivery of biotechnological discovery and innovation for the benefit of the society. Biotechnology will continue to provide intriguing challenges for the chemical engineers as the range, variety and extent of applications proliferates. Chemical engineers have the appropriate wherewithal to meet the surpass those expectations. The right combination at the right time in the right place. Chemical engineering is helpful in higher education, oil and petrochemicals including exploration, specialty chemicals and pharmaceuticals, food and consumer goods, utilities and environment, engineering consultancy and manufacturing, finance and management consultancy and other various miscellaneous fields. Now what is crop biotechnology? It has a dual effect of green biotechnology. The potential benefits where there is increase in the crop productivity, resistance to pests and insects, fighting plant diseases, increased food availability to the society. What are the potential risks? Health and environmental risks, socio-economic risks, and there is a competition of the biotechnological companies and the farmers. Molecular diagnostics is a collection of techniques used to analyze biological markers in the genome and the proteome. The technique is used to diagnose and monitor disease, detect risk, and decide which therapies will work best for the individual patients. So, molecular diagnostics offers the prospect of personalized medicine. Here you can see the old way of molecular diagnostics where the patient sample is taken and is observed under the microscope and the pathology report is given. The new way, patient's tissue sample or blood sample is taken and is put in a mass spectrometry where the proteins are detected and proteome mix is studied, and the DNA is studied by the different methods, and gene chip is created and a microarray image is obtained. In proteins we can see the proteomic image is obtained. Pharmaceutical biotechnology consists of the combination of the two branches which are pharmaceutical science and biotechnology. Pharmaceutical science can simply be defined as the branch of science that deals with the formulation, compounding and dispensing of drugs. Biotechnology can simply be defined as the application of the biological system, living organisms or their derivatives in making or modifying the products or processes for specific use. So the pharmaceutical industry is useful in many areas of manufacturing like the therapeutic enzymes, microbial polymers, anti-cancer drugs, phage display bacteriophages, excipients, amino acids, vitamins, minerals, carotenoids, microbial pigments, drug delivery system, immunomodulators, immunosuppressants, plant cell cultures, biofarming, animal cell lines, terrestrial organisms, marine microorganisms, antiviral drugs, antifungal drugs, antibacterial drugs, antibiotics, monoclonal antibodies, recombinant vaccine manufacturing and hormones, and diagnostic enzymes manufacture, adjuvants, and also in drug therapy. So the main contributions of biotechnology to vaccine development. Subunit vaccines were manufactured by the recombination of proteins in heterologous systems. Example, hepatitis B, acellular pertussis vaccine, HPV. Bivalent attenuated pathogens were used where the genetic manipulation for insertion of genes encoding antigens is done. For example, the manufacture of dengue fever vaccine, BCG, Salmonella, typhi, and adenovirus. The DNA vaccines were manufactured with immunization with recombinant plasmids. Example, the melanoma vaccine is manufactured. Animal biotechnology is useful in xenotransplantation, animal nutrition, animal vaccines, animal breeding, embryo transfer, transgenic animal production. Now, what are the different new vectors in biotechnology? Example, cosmids are plasmids that contain the cos cohesive terminus, the sequence having cohesive ends. They are hybrid vectors derived from plasmids having a fragment of lambda phage DNA with its cos site and a bacterial plasmid. Here you can see the image of cosmid vector. It has a polylinker, cos site, col E1 or E, where the site of origin of replication, and ampicillin resistant part. Bacteriophage vectors are viruses that infect the bacteria. The most commonly used E. coli phages are 1 phage, lambda phage, and M13 phage. Phage vectors are more efficient than plasmids. DNA up to 25 kb can be inserted into phage vector. Lambda phage is a temperature bacteriophage that infects the E. coli. The genome of lambda phage is 48,502 BP long, that is 49 kb, and has 50 genes. Here you can see the lambda phage. It has a Cl part, a replication gene, lysis genes, coat genes, the restriction sites having the lysogenic genes and the essential gene. Phagimid vectors are reconstructed plasmid vectors which contain their own origin and also contain origin of replication from a phage. P blue script SK plus or minus is an example of phagimid vector. Here you can see a part of the phagimid vector showing secretion signal promoter, the coat protein, and the displayed peptide. And the other regions are F1 ORI, dsDNA on, and ampicillin resistant part. Bacterial artificial chromosome BAC vector is a shuttle plasmid vector created for cloning large-sized foreign DNA. BAC vector is one of the most useful cloning vector in recombinant DNA technology. They can clone DNA inserts up to 300 kb and they are stable and more user-friendly. Here you can see the BAC vector, not 1 T7, hind 3 BAM H1, not 1 SP6, CM on S, Rep E, Par A, Par B. So all these are parts of BAC. Yeast artificial chromosome YAC vector, which is a plasmid vector, behaves like an yeast chromosome, which occurs in two forms, circular and linear. The circular YAC multiplies in bacteria and linear YAC multiplies in yeast cells. Here you can see the YAC vector, SEN4, sub4 occur cloning sites, URA3, telomere, BAMH1, HIS3, BAM1, 2, telomere, PMB1, ORI, ampicillin resistant part, trypsin part, TRP1, ARS1. Shuttle vectors. The shuttle vectors are plasmids designed to replicate in cells for two different species. These vectors are created by recombinant techniques. The shuttle vectors can propagate in one host and then move into the another host without any extra manipulation. Most eukaryotic vectors are shuttle vectors. Example of shuttle vector you can see, yeast origin of replication, leucine biosynthesis gene, yeast centromere sequence, multiple cloning site, antibiotic resistance gene, bacterial replication genes. Now M13 vector is single stranded DNA bacteriophage used as a vector for DNA sequencing. In this you can see the M13 6.4 kb vector with different parts including the ORI and the transcription areas and different G parts. So today in Principles and Processes of Biotechnology Part 5, we discussed applications of biotechnology, interdisciplinarity fields of biotechnology and new vectors in biotechnology. So thank you. Kindly subscribe, like, share and comment our channel Read MedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.